Okay.
Okay.
Yep. I think we're ready to go. We'll certainly have some people continue to join, but I think we're actually close enough to our starting time that we can actually begin. So I want to welcome this huge, robust crowd. I'm enormously honored and impressed and delighted to find that we have people who are joining us from all reaches of the globe. We have people joining from Israel, Europe, the UK, all across the United States, Canada, and I'm not even sure what time it is in Australia, but our reach goes to the middle of the night as well. So this is quite an extraordinary representation of the international work of the ML4 Foundation. So I'm honored. I'm really delighted, that we're here. I want to say that as sort of disappointed as I am that we're not doing this in person, because one of the things that's most valuable to me is seeing all of you in these every 2-year meetings. I think there's certain things that we can never replace by having that, and some of those are actually the clinical part of when Albert Misko, who you're going to hear from, our pediatric neurologist, gets to see your kids in person and conduct those examinations. We're finding in this strange world we're in that there are certain wins that we get at this time. And one of them is that we're seeing many more of you in this meeting than we probably ever would have. And in fact, there are many of you have asked your own personal physicians to join this meeting. We have scientists and clinicians joining from various hospitals. I'd like to welcome several physicians and scientists from hospital who I spoke with yesterday in Israel who are joining this newly, and just other physicians and caretakers who have not previously joined. So I'm excited about this. I will say that one of the things that this conference always represents, that to me as the head of the ML4 Foundation, and I'm Rebecca Overman in case I didn't introduce myself, one of the things that this represents as the director of the ML4 Foundation, that is a primary value of the ML4 Foundation, is collaboration. I think that one of the things that we do that is most valuable is that we bring the patients and the families who are experts in your children, and therefore experts in the disease, together with the scientists and researchers who are also experts in a different way in the disease, together to learn most about the disease, to create something meaningful for your children. And so for me, that's what today is about. And I really invite those of you who are listening to listen and learn. What you're listening to today is really an outcome from a great deal of what happened and was learned 2 years ago in the Atlanta meeting, where we learned an enormous amount from your children and from the research that was presented. And a great deal of work, an extraordinary amount of work, was done over the past 2 years, and you're going to hear about that. And I think more learning will be happening today and in the coming weeks after we talk about this, that will then kind of sail us forward in terms of where we're going to go from here. So with that kind of introduction, I'd like to introduce next Dr. Susan Slavenhaupt. Let me say just one protocol issue about how this is going to work today. As you know, you are all right now muted, not because I don't love talking to you all the time, but because we're blessed with many people on this call, so it will be easiest to keep you all on mute. We do want to hear your questions, however. So we ask that if you have any questions that you would like for the question and answer period that will follow each speaker, for you to use the chat button, which is at the bottom of your screen, and that you simply chat to me. So it'll say chat to Rebecca, and put any questions that you would like to be answered in that chat box. Those will come to me. And at the end in each question and answer, we will ask those questions. I will ask them on your behalf to the relevant scientist who was speaking. And that way we can manage everyone's questions, and Dr. Slaugenhaupt and I will manage the question and answer. By way of introduction, it could be a very, very long introduction. Some of you know Sue Slaugenhaupt personally because you knew her as the scientist who was early in the decades ago working to identify the gene that was causing the disease in your children. And Sue was one of the people who was credited with the discovery, co-credited with the discovery of that gene. And Dr. Slaugenhaupt's lab has been involved in disease discovery for years, ever since. Dr. Slaugenhaupt was co-credited with the discovery of that gene, the carrier test that has allowed for safe carrier testing since, creation of the first mouse model, which she generously has given to labs all around the world to enable other discovery and all kinds of interest in research for ML4, and has gone on to really make possible all other research in ML4 that has followed. Dr. Slaugenhaupt has had an illustrious career at Massachusetts General Hospital and is currently the scientific director of the Research Institute there, but still plays an extremely important role in guiding our research and has been an incredible mentor to Dr. Grishchuk, who has taken over primary responsibility for the preclinical research and has been an incredibly inspiring person, I will say personally, for me to work with. So I'd like Sue just to say a few words. Many of you know her and all of you should know about her.
Thanks, Rebecca. Thanks so much. Well, hello, everyone. It's really nice to see all of your faces in this Zoom meeting. I too wish that we were together in person, but recognize that if we had this meeting in person, so many of you wouldn't have been able to join us. So I think there are pluses and minuses in everything. And the big plus is that we are reaching you all around the world and that you'll all be able to hear from our scientists today and hear a little bit about the research going on in ML4. I guess my message for you today, and the thing I wanted to talk about for just a couple minutes, is a message of partnership. You are all our partners in research. You always have been, and I want you to understand how important it is that you feel like our partners. So many of you participated in our early genetic studies, and without you and your families and the parents and grandparents and other relatives of the kids, we never would have been able to find the gene that caused ML4. So many of you participated in those early studies. And now you have the opportunity to continue to participate by joining our natural history study and other studies that will be happening as we move towards developing a therapy. And it's so important, your participation. And so it's really great that you've come today to hear about the science, learn about the science, to understand what's going on in the laboratories and in the clinics. Because as we work to develop therapies for ML4, your participation will be crucial. I always like to talk to families about the fact that every time we hear about a new drug or a new treatment that comes online for a rare disease, it's important that you understand that that never would have happened unless people who had that disease participated in the research and participated in the clinical trials. And that's the only way it happens. And so you're our partners, and we're super proud to have all of you as our partners in research. And that's really the message that I wanted to bring to you today. And many of you I've known for as long as I've worked on ML4, over 25 years. And many of you I've just gotten to know over the last several years. And I really hope to have a chance to meet all of you in person at some point. So again, the message today is one of partnership, one of hope, and you really need to understand how important you are to all of the work that's going on. So Rebecca, don't know, did you want to go directly to the talks, or?
I'm just gonna have Randy Gold, the president
of the
ML4 Foundation, say hello, and then we can move to Yulia's talk, if that's okay.
Perfect.
Does that sound right, sir?
Yep.
Randy, are you all set?
Hi, everybody. Good morning or afternoon or evening, depending on where you are around the globe.
Or middle of the night
or middle of the night. Welcome to the first ever and hopefully only virtual ML4 conference. Of course, it's great that we have this method of communication, but it would be nice if we could have everybody together in person. I want to first thank Laura Willard and Dr. Rebecca Oberman for all of their incredible work planning not one, but two conferences. It is a mammoth undertaking to plan one conference, and planning a virtual conference actually has its own set of challenges. And these two folks have worked very, very hard to pull this off today. And they work hard every single day to move us closer to a treatment for our children. And without Laura and Rebecca, we really wouldn't be here today discussing these incredible new developments. So let's give them a virtual hand for everything they do for our organization. I mean, I can hear this applause. I mean, everybody's on mute, but I can hear it. So. Of course, we are so incredibly grateful to have the amazing team of researchers that you'll hear from today. Dr. Sue Slaugenhaupt, who just spoke, is such a special person, and she has been the primary force behind ML4, as you heard, for more than 2 decades. She started when she was 20, I think, 30 maybe, something like that. And Dr. Yulia Grishchuk has taken on the challenge of finding a treatment for our kids with the utmost determination and care. She has really, really worked hard to create a breakthrough for our children. And these two women take their work very, very personally. They know many of our children firsthand, and it's because of their work that a therapy is becoming a reality for our children. In fact, I want you to know that Dr. Grishchuk's work was so recognized as important that even when 95% of the laboratories at Mass General Hospital were closed because of the pandemic, Dr. Grishchuk's laboratory was one of the 5% that were allowed to remain open to continue the research that she was doing. That's the regard that she is held in the scientific community. Dr. Albert Misko, who we'll hear from, has examined more ML4 children than anyone else in the world and is working closely with Dr. Slaugenhaupt and Dr. Grishchuk to incorporate his clinical findings into the data that will be so important for regulatory approval for a treatment. And also today you'll hear from Dr. Brian Wishart, who along with Dr. Misko is helping all of us with the practical issues that our children deal with every single day. We really can't thank the 4 of you enough for the work that you're doing. Finding a treatment for a disease is absolutely God's work, and we all pray for your continued success. I also want to thank all of you who have put so much energy into sharing information and videos of your children. The questions that you ask and the answers that you share with our community are so helpful to all of us. And having a child with ML4 is emotionally and physically very, very hard. But together we find ways for all of us and for our children to thrive. So thank you to those of you out there who are working hard to raise money to support these 4 incredible researchers. We don't have a huge national foundation. The folks you see here on this call, all of you are what we have. There's no big black box somewhere with money just waiting to be put to use. So the work that you do, raising money for the foundation, keeps these researchers going for our children. So thank you all for joining today, wherever you are. I know you'll be inspired by what you hear. I wish all of us on the call today continued good health and safety, that we get through this pandemic quickly, and that we soon see the fruits of our labor and the researchers in the form of a gene therapy for our children. All the best to all of you on the call today. Thank you, Rebecca.
Thanks, Randy. I think now we'd like to turn to our first presenter. We're going to move to Yulia Grishchuk. So you've heard her name mentioned several times. Sue, did you want to introduce? I'm happy to.
Sure. Well, I would just like to say a few words, if you don't mind. Yulia is such a remarkable force, a remarkable scientist, and it's so exciting for me to have watched her develop this ML4 research program under me over the years and to move into a leadership position. You're all very lucky that we have Yulia at the helm, and the work that she's doing is really remarkable. Yulia is now an assistant professor in the Department of Neurology at Harvard Medical School and an investigator in the Center for Genomic Medicine at Mass General.
Thank you, Sue. So hello, everyone. I have to say it's a very, very special moment, and I'm so excited to be here with all of you today. I am going to share my slides and get started. Rebecca, can I ask you to unshare?
Yes, I am. Hang on one sec. Are you good now?
I am sharing now. I hope that now you can see my first slide.
Yes, Yulia, we can.
Yes, perfect.
Okay, so I'm really happy to give you an update on our gene therapy program today. And my talk today, this presentation, I built around those questions of what, why, and how. So in the beginning, I would like to tell you about this approach in general, just the very basics of it, what we're trying to do. And then I will try to explain why it's a good thing for ML4. And then in the end of my talk, I'm going to talk a little bit about what are the next steps that will have to bring us to the clinical trial. Okay, so let's start with the basics and just kind of discuss the terminology of gene therapy. So ML4 is a genetic disease. We all know that. And it means that it's caused by errors in a specific gene that in the case of ML4 is called mCOLN1. So those errors result in unfunctional protein and those are missing protein. And this leads to big consequences for the cells and organs. And in case of ML4, it's primarily the brain and the eye. So the logical thing to fix this error would be to replace the mutated, non-working gene with a working copy of it. And this is exactly what we're aiming on when we talk about gene replacement therapy. So we want to replace this gene that doesn't work with a correct copy of it, but often this working new gene also has some regulatory additional elements that added to this gene. And this is they're in charge of how much of this correct working gene will be produced in the body and where exactly it will be produced. So now we're talking about recombinant gene that will be replacing in the body the mutated gene. So now to deliver this working gene, we use viral vectors. And so these viral vectors, they utilize natural properties of viruses. And I'm sure that now we all know about viruses a lot more than we knew 2 months ago. So they utilize natural properties of the viruses to reach inside the cell. With exception that if we use virus as a vector, we remove everything that the virus uses to multiply and propagate in the body. So in the end, we only keep the shell that uses the power of the virus to get inside the cell, but we fill this shell with the gene we want to get produced in the body. So most of the viral vectors that are being used in biomedical research and as drugs now are based on adeno-associated viruses. What do we need to know about this virus? We need to know that this is a non-pathogenic and harmless virus that is used to deliver, this genetic information, and it has a capsid. And so this capsid can be different for different types of adeno-associated viruses. They're also called serotypes. And so those capsids, they define the properties of the virus that make them work for different cell types and different organs. Some serotypes, for example, when we talk about delivering this recombinant genes to nervous system, we often use AAV9 type of AAV. When we talk about targeting it and delivering to liver or systemically, we use AAV8 serotype or type of AAV. There are other types as well. So if we now put it all together, so we have a missing or misfunctioning gene, and we use adeno-associated virus to package or insert the proper copy of this gene to be delivered to the organ that is affected or suffering in this disease. So I also put together some of the links for you here that are from FDA or American Society for Gene and Cell Therapy or Rare Diseases Association. And so they have also some kind of useful information about different approaches in gene therapy, or also about more details about this approach that is called in vivo gene replacement therapy. So now why we think this is the great option for ML4? So this is based on the fact that we're dealing with a genetic disease, and it's kind of a common sense, right? So it's the best way of dealing with this disease is to fix the root cause, to replace this gene, rather than chasing one by one secondary consequences of the loss of this function, the loss of this gene. And so this is true for any genetic disease, and the field was developing to make it reality, this type of therapy reality for genetic diseases for a while. And there are recent breakthroughs. So in the last couple years, there have been 2 approvals of gene therapy drugs in the US. But actually, the first gene therapy drug, which called Glibera, was approved by European Union for the treatment of lipoprotein lipase deficiency, even in 2012. So it had great safety profile and was discontinued for production in 2017 simply because there were no demand, and most of the patients with this rare disease already obtained this treatment. So in 2017, a first drug for a retinal disease was approved in US, and then a year ago, first CNS drug was approved, and this is the drug for the treatment of spinal muscular dystrophy. So with this breakthrough and this tremendous success, I have to say that there are a lot of ongoing and active trials testing AAVs for various diseases. So as of now, there are 211 trials registered in US, and 46 of this testing gene therapy for the brain diseases. So with this number of trials, there are kind of of advanced programs, and the number of new drugs and new approvals are expected in the next couple years that expected to kind of be curative for types of hemophilia, Pompe disease, Huntington, spinal muscular atrophy, different form of it, ALS, and mucopolysaccharidosis. So we obviously would like to kind of ride this wave and use advantage and use all that field has worked out for ML4.